This is the first of 3 lectures on the special senses. We're going to start with an overview of what the senses are and how they're classified, and then we'll focus in on the special senses. So first of all, what's the difference between the special senses and the other senses? The other senses are referred to as the general senses. General senses have sensory receptors found scattered throughout the body. They are subdivided into 2 groups, the somatic senses, of which you're conscious. This includes receptors in your skin, such as touch, temperature, and pain receptors, your sense of awareness of your body parts, which is called proprioception, basically telling you from receptors in your joints and muscles where your body parts are and how they're moving. And the other category of general senses is the visceral or subconscious sensations. These are things you're not usually conscious of, such as your blood pressure, how much oxygen and carbon dioxide are in your blood. Stretch in hollow organs is something you may be aware of. If it's something that has to have some kind of influence on your behavior, such as if your stomach is really already stretched out, maybe you should quit eating, or if your bladder is stretched, then you probably need to go empty it. But there are other hollow organs that you don't have much sensation of, including the ureters and the intestine. In general, the only time you can feel those is if they happen to move against the body wall, and then you're conscious of the sensation of the body wall more than you are the hollow organ. So that's all we're going to say about the general senses. We're going to focus on the special senses. Special senses are all somatic, and remember that means that you're conscious of them. And the special senses are special because they have localized receptors that in general have some kind of complex organ structure, and as it happens, they're all located in the head. Of the special senses, 4 of them are going to give you information about the external environment, and 1 provides information about the internal environment. So our learning outcomes from the chapter are to describe the sensory organs of smell and taste. Those are the 2 special senses that we're going to be working on in this part of the lecture on the senses. So we're going to look at the olfactory pathways and how they work, and the gustatory pathways and how they work. So of our 5 special senses, olfaction is a chemical sense. You are sampling chemicals in the environment that you inhale through your nose. Gustation is also a chemical sense. You're sampling chemicals in the food that you eat. Vision is a light sense. You're actually sensing through photoreceptors. You're sensing photons of light that hit your retina. Equilibrium and hearing are both in the ear, and they are mechanical senses. That is, you're actually pushing in some way on the receptors, either from sound waves or from rotation of the head. So let's focus in on smell or olfaction. You have olfactory organs, if you want to call them organs. It's really more of a layer of tissue located up in the superior nasal cavity on either side of the nasal septum, way up high in your nasal cavity. There's just a thin layer of olfactory epithelium, and embedded within this olfactory epithelium are olfactory receptors. The receptors themselves are actually little cilia with sensory receptors embedded in the membrane of the cilia, as we see. So here you are looking at a parasagittal view of the right side of the head. The nasal septum's been removed. And what you can see is here are the olfactory receptors, that is the little sensory cilia up in the upper nasal cavity. The olfactory epithelium itself, as you can see, is up above the superior concha, which is the uppermost of the 3 conchae of the nasal cavity. And the olfactory nerve fibers run through the olfactory foramina, the holes in the cribriform plate of the ethmoid bone, and they impact on the olfactory bulb. The olfactory bulb is actually part of the brain. It's on the surface of the brain, and within the olfactory bulb you'll have the synapses of these nerves. From the olfactory bulb, the axons of those neurons will pass through the olfactory tract, and from there they're going to go to different parts of the brain. So when the odorant molecules, the little chemicals that you've inhaled, reach the olfactory cilia, if they bind to those cilia, and you may have some odorants that bind to the cilia of one of these cells and different odorants binding to the cilia of a different cell, then it is going to open some voltage-gated channels or some ion channels, and that's going to create a nerve impulse along the axon of that neuron that's running up through the olfactory nerve fibers. The rest of the olfactory epithelium consists of some supporting cells. You have olfactory glands which just secrete a little bit of a fluid, a watery fluid, to help dissolve the odorant molecules in addition to the receptors. So the supporting tissue of the olfactory glands is simply there to make sure that the olfactory receptors are actually going to be able to do their job. So here we're seeing a close-up of the membrane, and this is kind of upside down of the previous view, because here you see the mucus layer at the top of the picture and the cell at the bottom of the picture, which is, as I said, upside down from the way the odorant molecules are inhaled. They're actually inhaled from the lower parts of the nasal cavity up, and here you're just looking at it upside down. So the odorant molecules attach to their receptors, actually an enzyme which activates the conversion of ATP to cyclic AMP, and cyclic AMP at the other end of the cell is going to open some sodium channels. As the sodium enters, that starts a nerve impulse, and so you can see the nerve impulse here. This slide is giving you more information that I'm going to test you on. It's just nice to see that full picture, but basically the bottom line is when the odorant molecule attaches to the cell, then it starts a nerve impulse. So again, this view's upside down, so you see the stimulus coming in to the dendrites, those sensory cilia, and you're going to see here on the axon, you're going to see action potentials occurring when the odorant molecules attach. Again, too much information. Okay, so following that nerve impulse, once it runs through the olfactory nerves, it's going to get to the olfactory bulbs. Remember, the olfactory bulbs sit on the ventral surface, the bottom surface of the brain. Should have seen that when you studied the brain last semester. From the olfactory bulbs to the olfactory tract, and please notice 1, 2, 3 destinations for olfactory sensation. The olfactory cortex is part of the cerebral cortex. That gives you your conscious awareness of what smells you are smelling so that you can analyze and identify it and name it. The hypothalamus connects to the visceral body functions. This may stimulate the digestive tract for getting it moving if you're smelling something that's food-oriented. And the limbic system is the center of mood and emotion. So here is where olfaction is tied to changes in your mood. It may make you happy. If it's an unpleasant odor, it may make you unhappy, feeling nauseous or repulsed by the smell. It depends on what you're smelling. So you've got those 3 information centers that you reach. And this is notable, the fact that the olfactory sensations do not go through the thalamus, because olfaction is the only sense that doesn't go through the thalamus. All your other senses, vision, hearing, they all have to go through the thalamus to reach consciousness. Olfaction is the only one that gets to consciousness without going through the thalamus. Okay, what do we mean by discrimination? Discrimination is the ability to tell one thing from another. So think of two things that are very similar but that you can tell the difference between. And remember that your sense of taste is actually based on your sense of smell. So can you tell the difference between two closely related nuts or spices? Can you tell the difference between pecans and walnuts? Can you tell the difference between butter and margarine? That's the kind of olfactory discrimination that your sense of smell gives you, and the way it gives it to you is by having different combinations of odorant molecules. There are probably thousands of odorant molecules that we can discriminate. Now, the other interesting thing about your sense of smell is that those olfactory neurons actually don't last that long. These cells are in contact with the external environment, so their lifespan is kind of shortened by that. But you do actually produce new olfactory neurons really throughout your life. And unfortunately, as you get older, the rate of replacement of olfactory neurons declines. This actually has a clinical application. It's a pretty well-known phenomenon that geriatric patients don't have much of a sense of smell, and it affects their willingness to eat. If the food doesn't taste good or they can't taste it, then they have less enjoyment of food. And so what you find is that older patients have to be coaxed into eating because they just don't appreciate food the way they did when they were younger. Okay, moving on to your sense of taste, your gustatory sense or sense of gustation. Taste receptors are found definitely mostly on the tongue. Some of the receptors are found in the pharynx or throat, the larynx, even down in the lungs. You have some receptors for bitterness, and in the stomach as well. We're going to focus on the ones that are found in the tongue, and those are clustered into taste buds. So a taste bud is a little collection of cells associated with the papillae. Now, don't get a papilla confused with a taste bud. The papillae are the little bumps on your tongue. They're called lingual papillae. Remember, lingual means tongue. And so they're found on the surface of the tongue. So papillae are the bumps on your tongue. The taste buds are kind of embedded in them. We're going to stop for just a second and look at the kinds of papillae. Filiform papillae are the majority of the bumps on your tongue. They do not have taste buds. Just the fungiform and the circumvolate. The fungiform papillae are found over the surface of the tongue. The circumvolate papillae are only found on the back of the tongue. So we'll take a look at that in just a minute. Each taste bud, again, has the sensory cells plus some supporting cells. The gustatory cells, like the olfactory cells, have cilia or extensions on the surface of them. But the gustatory cells are not true neurons. They're specialized epithelial cells that have a connection with neurons at their base. The cranial nerves that connect to the gustatory cells, there are actually 3 different cranial nerves that connect. There's not just one nerve for your sense of taste. And those cranial nerves are going to send impulses to the thalamus. Remember, everything except smell goes through the thalamus. And then to the primary sensory cortex, which is in the parietal lobes. There's a gustatory center on each parietal lobe, right and left, to help you interpret what you're tasting. So here we see the surface of the tongue. You've got sour, bitter, salty, sweet. There is another taste that is associated with the tongue. This is a fairly new taste called savory, or the actual proper name for it comes from the Japanese umami. And the reason that umami is the proper name is that the Japanese researchers were the first ones to actually identify the receptors. This idea that you have different tastes on different parts of the tongue is no longer accepted. They're pretty well scattered. So here you see umami, and this, I think, picture is mislabeled. What you're seeing here in these large circles in the back, those are the circumvolate papillae. and they primarily contain bitter receptors. You also notice you have water receptors. There are definitely water receptors found in the mouth and throat, and they just provide you feedback that you're drinking water so that you quit drinking water. You've slaked your thirst before you've overhydrated. If you waited until you'd actually absorbed that water into your bloodstream before you stopped being thirsty, then you'd drink more than you need. Okay, so here we see a close-up of a circumvallate and a fungiform papilla with an extreme close-up of the taste buds shown in the side. So let's look at those taste buds. Here you see a micrograph of them, and here at the bottom you see the little taste cells with the taste hairs, which are actually microvilli, not cilia. And of course there has to be an opening in the surface of the tongue. Notice that these are on the sides, not the tops of these papillae, so that the odorant the taste molecules have to be dissolved in saliva and diffused down to reach the pores. Down at the basal end of the cells, you can see kind of plugged into the bottom of the cells, the neuron is attached. So again, here's our view showing the stimulus is going to come here. The taste molecules, whether it's salt or sugar or amino acids or whatever, is going to hit a receptor cell that's specialized for that particular category, salt or sugar or whatever. Here is the neuron, so that when the receptor cell gets stimulated by the taste molecule, it starts an action potential, a stimulus potential, that simply transmits the electrical signal to the neuron. And so you don't get an action potential on the receptor cell, you can only get an action potential on the sensory neuron. This is the axon of the sensory neuron, and of course you'll remember these guys from A and P1. Those are Schwann cells, which you remember provide the myelin sheath for neurons in the peripheral nervous system. Okay, so again, we're looking at an extreme close-up of the membrane, and we're seeing how the acid or the salt or whatever is going to hit the receptor site. That's going to open a gated channel, which is going to allow sodium ions to enter the cell. That depolarizes the membrane. And that works for salt and sour receptors. For sweet, bitter, and umami, you've got G proteins. This really is still too much information for you to remember for the test. If you understand that when the taste molecule hits its receptor, that starts the flow of ions, which then gets transmitted as a nerve impulse, then you're in good shape. You know everything you need to know. Okay, so for primary taste sensations, again, this is out of date. The fifth one is umami, which is savory. So sweet, of course, is for sugars, salty for salt, obviously, sour for acids. Bitter is mostly bases, and things that are bitter tend to be poisonous, which is why we could associate bitter with unpleasantness. If you taste something and it's bitter, you probably shouldn't eat it. And of course there's some bitter things that we choose to eat that we know are safe, but if you were like stuck in the wilderness and looking for something to eat, then you wouldn't want to eat something that was bitter, definitely. Okay, and then savory is the ability to sense some particular amino acids. So you have the ability with your sense of taste to tell the basic nutrient groups that you need. You've got your sugars, you've got your amino acids, you've got your salts, and then the ability to tell if something's acid or base. Okay, so the way your author does it, he puts umami as a separate characteristic, but in fact it is really just another one of the basic sensations, and we talked about warmth receptors. All right, gustatory discrimination, again, remember that's the ability to tell specifically what something is. Again, G proteins, chemically gated ion channels, all of that is just too much information to test. So it's the end result we're after. We're going to get action potentials, which is a nerve impulse. Those nerve impulses are going to run through to the brain. This is just kind of an interesting little quirk. It's the ability to taste a particular chemical. I think you're actually going to do this in lab. I'm not 100% sure that they actually still do that in lab. But this is just sort of a little quirky genetic difference that some people can taste a particular component called PTC and other people can't. But it does point out something that you've probably experienced, that you may be able to taste things that other people can't, or vice versa, they can taste things that you can't. So it's just something to be aware of. And again, taste buds decline, so older people tend to lose their sense of taste. Okay, so one thing that's not in here and should have been is the pathways to the brain for gustation. So let's talk about them very briefly. You've got 3 cranial nerves that carry taste sensations. The facial nerves, that's cranial nerve 7. And the facial nerves carry sensations from the anterior 2/3 of the tongue. So that's going to be predominantly most of your taste sensations. The glossopharyngeal nerve and the vagus nerve also carry taste sensations, but more from the back of the tongue, the throat, and those are going to be primarily your bitter taste receptors. All of those sensations have to go to the thalamus, from the thalamus to the gustatory cortex, which is in the parietal lobe of the brain, and that gives you your conscious awareness of taste. Okay, part 2 of this lecture series on the special senses will cover vision. So I'll be back with that one soon.